As many as 100 billion cells in the human body die daily, and are replaced by new ones, so it's not that bad. This is effective in maintaining stable growth and preventing our weakened cells from forming into diseases. Mitosis is the division of one cell into two daughter cells with duplicate DNA. But how exactly does that happen? As the procedure is gone through, remember the acronym IP-MAT-C. Interphase is the phase in between divisions of the cell. During interphase, the cell is growing and preparing for the next division, but it's mostly doing its regular job, or its metabolic activity. So for example, muscle cells are contracting and relaxing, liver cells are detoxifying, stomach cells are digesting, nerve cells are firing, and you get the gist. Both the nuclear membrane and the nucleolus are still clearly visible in the cell, and the DNA within the nucleus is in the form of regular, uncoiled chromatin. When the cell begins preparing for the next division in interphase, the DNA, as chromatin, is copied. So during interphase, the cell is still preparing itself for mitosis, but then it moves on to prophase, which is the actual first stage of mitosis. So during prophase, the DNA, still as chromatin, coils into distinct chromosomes now. Each chromosome contains two chromatids, or copies of the same part of the DNA, attached at the centromere, or you can think of it as the vertex point. The nucleolus and nuclear membrane then break down, releasing the chromosomes into the cell. Two dense areas called centrosomes appear at opposite poles of the cell. This is important because the centrosomes contain two cylindrical bundles of microtubules, which are the centrioles, that direct spindle fiber construction, which is important in later phases. But for now, just know that the cell starts building spindle fibers so then they can send them out to perform functions. Frick.
In metaphase, which is the second phase of mitosis, spindle fibers made of microtubules stretch from the centrioles and attach to the paired chromosomes at their centromeres. Because of this, the chromosomes line up along the middle of the cell, called the metaphase plate, and become easier to identify. In the third phase of mitosis, anaphase, the spindle fibers that the centriole sends out pull on the microtubules to separate the chromosomes. The chromatids separate and move slowly until they are at opposite poles of the cell. After chromatids separate, they are considered individual chromosomes. Chromatids reach the opposite poles of the dividing cell, telophase begins. The spindle fibers detach and the chromosomes return to their chromatin state. New membranes form around the chromosomes to create the new nuclear membranes, and the nucleolus forms in each of the newly forming cells as nuclei. Now that mitosis is completed, the final stage of cell division, cytokinesis, begins. The plasma membrane begins to pinch down along the middle of the cell between the two new nuclei. The part that pinches down is called the cleavage furrow. The cleavage furrow's pinching splits the cytoplasm into two parts for each of the daughter cells. This completes the process of division.